Okay, I think we're going to get started. Thanks everybody for joining and bearing with us when we found a place for pizza. Today we have the pleasure of Sarah, who's going to be giving us a talk about discrete diffusion in sequence space. And she spent the past couple of years at MSR working on a project just like this. So I think we have a pretty good talk today. Before that, she actually did her PhD at the University of Washington, not with David Baker. Not with David Baker.
Great.
And so with that, welcome Sarah, and we look forward to your talk. Thanks so much.
Awesome. Thank you for that. And thanks for the nice intro. And thank you everyone for coming by today. It's great to be able to share this with you. So to start, really our motivation here is kind of this idea of using biological molecules to unlock new capabilities. And we've seen, number of examples explode doing this in the past few years. So for example, creating molecular tools. Right here, I'm showing an image of green fluorescent protein. We saw ESM3 be released just a few weeks ago, who had just designed a new GFP. Similarly, we are interested in proteins that can perform new chemistry. So here's an example of an enzyme, I believe out of Francis Arnold's group, which was engineered to catalyze this reaction that had not been catalyzed before. And alternatively, we're also interested in designing therapeutics. So kind of a main driving force behind these is designing protein molecules like antibodies and whatnot. So a question that we're really asking and that motivates us here is, can we design novel proteins that expand this functional space? Oops, my cursor is not working. And to do that, we need to think about this protein engineering problem that we describe here. So the question we're really asking is, given a desirable function, is there a particular protein, which is in simplistic terms, just this string of amino acids that will give us a desirable observed function. And to solve this, we first need to understand what sequence would give us those functions that we already know and already exist. And to solve this, we know that often, but not always, this relationship between sequence and function is mediated by a 3D structure. And this is what we call the sequence structure function paradigm of protein engineering. Now, historically, and with many of these models that we've seen, protein design has been addressed in reverse of this natural paradigm, using structure-first approaches. And this has worked quite successfully in solving problems like protein binders. We've seen kind of an explosion of examples coming out of RF diffusion's model. And in this scheme, what we do is first identify a function of interest. We then identify some intermediary structure or motif that we're interested in scaffolding or building a binder for. And then we use alternative models to then predict the inverse folding sequence. So now that we've designed a protein, how do we inverse fold that to a corresponding sequence, which we can then express and test in a lab? And so in our model, in our work, we propose that in fact, sequence and not structure is this universal protein design space. And ultimately that it's desirable to go from a desired function directly via a protein's sequence. And in doing this, we can bypass some of these major challenges that come with structure-based design. First, we eliminate the need for this indirect sequence design of going from protein structure to sequence via an additional model. And similarly, we have access to many more sequences than structures. So this is just a depiction of the possibilities of combinations of amino acids that you can make that compose a protein. Only a small set of these are saved in databases like UNIREF50. And an even smaller amount of these have been crystallized as protein structures and are available. And lastly, we hypothesize that our sequence-based approach can now handle disordered regions. So with this in mind, we take inspiration from biology and ask if we can use this information that's naturally encoded in protein sequences to train models which can generate novel proteins and thus expand our accessible functional space. So to that end, we developed EvoDiff. This is an evolutionary scale discrete diffusion model that operates over protein amino acid sequences. So if you're not familiar with diffusion models, you take an input sequence at time step 0, which I'm circling over here on the right, and then you iteratively corrupt it over t time steps until you reach something that's been properly corrupted or resembles noise. Now, in this EvoDiff model, we evaluate a few different approaches. The first here is called order agnostic autoregressive diffusion, or OADM. This was a model proposed in ICLR in 2020. And in this scheme, your corruption is that of a mass token. So you iteratively corrupt every position in the amino acid sequence in an order agnostic fashion, so effectively randomly, until you achieve a sample that has been completely masked. Oops. Oh, okay, I'm going to manually iterate through this example here. And so what you can see is that we're iteratively masking tokens. Our model is learning to predict these tokens with context from both directions. And we do this over multiple time steps until this model then can learn how to unmask these various tokens. At inference time, we then decode starting from an all-mask sequence and iteratively unmask each amino acid residue in a sequence of set length. Okay, so that's one scheme that we evaluated. This second scheme is called discrete denoising diffusion probabilistic models, or also known as D3PM. And in this corruption scheme, now our individual amino acids are mutated to other residues by sampling these transitions according to some transition matrix. In our work, we explore two different types of transition matrices. These are known to play kind of a significant role in the performance you can get out of a model. But here we look at a uniform transition rate, where every amino acid has an equal probability of mutating to a different amino acid. And second, we use this blossom transition matrix, where we use evolutionary homologs of other protein sequences to select particular mutations that are related as we corrupt to a fully noise state. Now, in this case, our neural network is trained to undo this corruption. However, this is a much more challenging task because whereas before our model knew the positions of the amino acid tokens it was trying to guess, in this case, it both has to figure out which mutations exist in the sequence and then learn to correct those. And so together we have 3 models and 2 discrete corruption schemes that we evaluated in this work. Overall, we found that our mass corruption scheme did quite a bit better than the D3PM performance. This was similar to performance we had seen on, or that the papers had reported on text data. And so we'll continue this talk noting that EvoDiff-seq is an OADM protein diffusion model. Okay, I want to take a moment to acknowledge that EvoDiff generalizes both masked and autoregressive language models. In this instance of OADM, we're learning every order of denoising at every step, meaning at every position of our amino acid sequence. This is kind of similar to what we call a left-to-right autoregressive model, in which the model is learning to predict the next token based on the previous tokens. However, this is only one decoding order. And of importance here is this kind of limitation in what you can do at inference time, in that you can only decode in one order, not allowing you to conditionally generate points in the sequence arbitrarily. And we'll circle back to this later in the talk. Second is this comparison to a mass language model like CARP and ESM. These models are effectively learning one step of our OADM model, and that they're learning one unmasking order for every protein sequence. And we see that that results in better generation at lower percents of masked residues, as it should, but it does not enable generation of sequences de novo without some additional tricks to help that. Okay, and so we did all of these comparisons. We train on our model on UNIREF 50 sequences. This is 50 million protein sequences that spans many organisms and many functions. And to compare all of these models, we also train an autoregressive and use mass models from the literature. These are all trained on 640 million parameter convolutional neural networks. Great. And so an example of what then happens at inference time is given by this nice schematic here, where we start with this all masked sequence of a fixed length, and we can iteratively decode amino acids within this sequence until we get now a new folded protein. And we can visualize the result of that process, both in sequence and structure space. Okay, so now that we've trained our model, we want to show that EvoDiff now enables controllable generation of plausible and diverse proteins. Yeah, go for it.
So just to make sure that I'm following what it is you're doing, it seems to me a little bit like, I'm very familiar with the model protein,
Yes.
scratching that network. And it seems like the training objective is basically the same, except you throw away the protein group. And you train on more examples because you have later sequences. Is it is that really what's happening? So essentially you just, in your training of a model like protein MPNN, everything's teacher forced, and you're just you pick a random decoding order and then you give it everything that has been sort of decoded up to that point, and as a condition, and then you have it predict the next residue in the decode. And that's like one training example. It seems like that's the same thing here, where you're not trying to unmask multiple tokens at once, you're just unmasking one at a time.
Yeah.
If that is the case, I'm sort of wondering, is there anything conceptually different here that from the protein MPNN model, other than not using correct?
Yeah, I'm not. Oh yeah. So the question, or well, you probably heard he has a mic to repeat, is kind of how similar is this training task to protein MPNN? I'm not familiar with the exact way that they train the sequences in protein MPNN, but one thing I want to call out is that one training step for us is picking a time step, which would be a number of residues to mask, and then tasking the model with unmasking all of those at once. Yes, and then you do that iteratively over many time steps, meaning the length of the sequence, and so then you have infinitely more training steps in that way. Does that make sense? Which I think is slightly different from how you describe MPNN. Okay, cool. Yeah. Oops. Let's speed her up. Okay, and so now we're going to go through the plausibility, diversity, and kind of some fun tricks about how we can conditionally design new sequences here. Okay, first, to evaluate the plausibility of the proteins that we generate here from scratch and from sequence alone, we ask our model to generate a sequence for us. We pass it through a structure prediction model. We've done it with both OmegaFold and AlphaFold. We task a separate model to now inverse fold this structure into a new sequence. And we define this term kind of point from these structure-based methods called the self-consistency perplexity, which is describing some similarity of two sequences against this similar predicted structure. Now, if we take some sequences from our test set, so that UNIREF set of real protein sequences, we see that by mapping the self-consistency against the PLDDT, there's a number of sequences that fall into this lower right bound range here that we're highly confident in and that are very self-consistent. But even within the test set, we know that these methods aren't perfect, as we have kind of a number of real proteins that are not captured by this metric. But this is sufficient for us to do some comparisons. Now, if we look at some of these examples produced by EvoDiff, we see that we capture a general distribution. And this is great because our model is quite small. It hasn't been trained that long, and this is quite a good result for us. We can pull out some of these protein sequences from this lower right quadrant here, and we see that across a range of amino acid lengths, we get some reasonable looking structures with high confidence. We're now in the process of testing some of these in the lab, and we've got some preliminary results that I'm not ready to share quite yet, but are quite exciting. Okay, now we can compare this to some of our baseline models. Here's our sequence-based diffusion. We can compare this to our left to right autoregressive model. We trained this from scratch using same architecture, same data set. It does quite a bit better. This is the state of the art, and it's a much more efficient training task than the one that we're learning on here. But we do pretty on par with the SOTA. Second, we generate a bunch of sequences from ESM2 from scratch. And we see that these are, without these kind of tricks to get them to generate nice, clear proteins, it does quite a bit worse than a generative model. But we expect this. These two examples on the left are generative training tasks, and the one on the right, yes, right, is not. Okay, so hopefully I've convinced you that we've trained these models well and they can generate plausible sequences on par with SOTA. But next, we really wanted to look at the diversity of these generated sequences. Okay, so to do this, we start with a set of test sequences. We pass them through a separate model, PROT5, and take these embeddings and map them in a 2D UMAP space to just kind of have a visualization for this. And we repeat this same step with generated sequences and map them in those same space. Here we can see that we have fairly good coverage. If you're like me, you're probably not convinced of this because there are some regions missing here. Still looking into this, don't know what these are, but overall we do okay. And now we can quantify how well these two distributions of data fit using this FPD metric. We coined the Furchette protein distance, where a low number is going to indicate better fit and a higher number is going to indicate a worse fit. Now, again, if we compare this to our SOTA model, we see that, okay, it does a little bit worse than our state-of-the-art model, and that can capture some of these missing regions, but still doesn't do great. But keep in mind that we're interested in EvoDiff for that capability of conditional generation, and that we have more flexible abilities at inference time. Second, we can compare this to that protein mass language model, again, not a generative task, and it does fairly poorly at this particular evaluation. And lastly, we can compare this to structure-based diffusion methods. And so again, we repeat this, we generate a bunch of, we inverse fold a bunch of sequences from generated RF diffusion structures, and we see that we don't get that coverage. And we attribute this to this idea that we're training a model at evolutionary scale. So it must be learning something about all these various examples. Okay, and so to further kind of investigate what's going on here, we can run another comparison where we'll take those sequences, run them through a PROT-trans model, which will give us some fractional estimate of the secondary structures within those sequences based on sequence alone. And we can map those onto a 2D space. So this is now just plotting the percent of helix present in those sequences versus the presence of strand. And if we just take this from UniRef, that map looks something like this. Now, if we produce our sequences, again, we see these kind of two regions of high helical, high strand propensity, as well as this region right here. And we can compare this to a few different baselines. Again, that LRAR, we do a pretty good job of recapitulating that. ESM2, again, does quite a bad job. And interestingly, RF diffusion, while it does a great job at capturing these secondary structure elements, it's kind of missing this region right here, which is that of disordered or unstructured proteins. And it's been kind of a little while since we put this out. And now we know this, that we've kind of learned that these structure-based models tend to over-approximate secondary structures or, be inclined towards generating highly structured motifs, which makes sense based on the tasks that they were trained on. Okay, and so to conclude, we can say that EvoDiff generations capture these secondary structure elements that mirror natural proteins, and that we're capturing all of these secondary structure elements. And now, hopefully I've convinced you that our generations are diverse, But we also really want to talk about this interesting feature about OADM, which is this idea of conditional design. Now, in this, I've discussed this a few times already, but in our conditional protein generation, what we're doing is fixing some subset of a protein that we're interested in and in-painting or resampling the remainder. And we do this in a random decoding order. We can do this for 3 various tasks, which I'll show here today. The first is this idea of evolution-guided generation, where we use a sequence alignment to help guide those generations, sequence in-painting, as well as functional scaffolding. So let's get into this first example here. Now, to do this, we have to describe an additional model that we trained that I did not mention earlier. But we actually trained a second model, which we call EvoDiff MSA, using that OADM task. This leverages evolutionary information of proteins. So given a query, we'll subsample up to 64 sequence, multiple sequence alignment that contains homologous sequences. We then train this model in the same fashion that we trained our EvoDiff-seq, but now over many sequence alignments, iteratively corrupting until we have a fully masked MSA. In this case, we now use 100,000 MSAs, which were available from OpenFold at the time, which again cover this large organism and functional space. And this time we actually use the MSA transformer as the backbone, which is about 100 million parameter model. What's cool about this is that it's effectively a local, an amortized local generative model, in that within a single MSA, you're able to capture information about protein families and thus generate new functional proteins with that information. And so we can discuss this first task with that in mind. Say we have a particular family that we're interested in generating a protein for. We can construct an MSA of that desired function. We can then mask out this query sequence and using our model, regenerate the query sequence, which is now, in hope, a new protein sequence. Okay, and now we can hopefully generate, and now we want to kind of validate that we're able to do this. So we take that original MSA, we mask it out, pass it through the model, and generate a new query sequence that now corresponds to that family. We can use this same workflow to then predict its structure and its self-consistent perplexity and get the same plot here where we're looking at the plausibility of a sequence as being in this lower right quadrant here. If we map our EvoDiff MSA generations on top of that, we see we do pretty well and that we're able to get a high number of sequences that fall into this lower right quadrant here. Oops. I will show you the results of those in a second. But what we can also do is kind of find a match for that family by taking the original query, predicting its corresponding structure, and then evaluating its TM score. This is going to tell us how close in structure that generated query might be to the original query sequence. And overall, we see that we can conserve a good amount of structure in that they're more similar, but more diverse than comparisons using ESM MSA itself or something like a POTS model. And again, we see a good amount of sequence novelty in doing this approach as well. We see that our generations are overall novel, but still preserve that desired structure. And now we can pull out a handful of these examples. So on the top here, we're looking at those generated queries colored by their confidence score. And under it, we have that corresponding true query or originally query sequence and how it performed in AlphaFold as well. And we can see that we have fairly good correspondence between the two visually, but also numerically. Great, moving on, we can tackle a second task. This is, yes, go for it. Yeah. Ask away.
When you're predicting structure,
yes. Actually, yeah, so for these we used omega folds because of the, yeah, I think I misspoke and said alpha folds. But these are actually generated using omega fold because we didn't want the contrast from that sequence alignment, obviously. Yeah. Yes, yeah. Hi.
It's a fun question.
Yeah.
Hypothesis. Why the sequence asking has actually worked quite well. Because the sort of data that you actually have allows you to approximate roughly 1D or 2. Really
Yeah, this is a great question. It's to repeat for the audience or the online audience, asking if like why we're able to achieve this capability by just masking the query sentence sequence compared to a structure-based method. I think, and I'll show in a little bit, the strengths of the two models are quite orthogonal, and there is definitely a path forward where, you know, both of these modalities can integrate and be stronger together. Why we might do better or are able to do this at particular tasks, especially for the MSAs, is the MSA data is like quite a bit more structured than this sequence data. So it's learning a pretty clear task. We've seen, you know, from AlphaFold that MSAs can help with protein folding. And so it's not surprising that if you start with something that's quite folded, you can predict a sequence that will fold into something similar as well. Yeah.
When, let's say, testing on whether or not just the sequence asking like led to similar results just with MSA, or like is it doing something that resembles like learning evolutionary information just with like sequence?
Repeat this.
Yeah, I guess like what I'm curious about is like, is the sequence masking that you're doing somehow over time like learning evolutionary?
Yeah, we could do some analysis on that. That would be interesting to like look at to create like a substitution matrix based on our generated sequences, maybe. Is that kind of weird? I don't have any thoughts on that is the answer, but this is an interesting thought. We were hoping to kind of impart this knowledge into one of our EvoDiff models, the D3PM model, by using that Blossom matrix. We were like, oh, maybe if we give the model like hints into how it should evolve, it can generate better examples. And that did not help. It ended up generating like very ordered helices like most of the time. So yeah, maybe it's learning that on its own.
On the slide.
Yeah.
When you say something like 0.67% similarity on the bottom there.
Yes.
What is that to? I assume it's like sequence identity is something.
Yeah, we only did sequence identity of the generated sequence to the original query. We did not try and look at it at every sequence in MSA, which arguably might make more sense. Yeah. If you raise your hand, I'll try to get the mic to you. I'll try and look out. Oh.
I guess that's where I'll have the question.
Yeah.
Actually, if you go back to one slide, kind of curious. So some reason the POTS model is not doing so well compared to.
Yeah.
My question is related to the POTS model. Okay. My question is related to the POTS model. I'm wondering if you have some ideas. What's going on with the POTS model? Why is it not able to generate? If I understood correctly, the POTS model is fitting on the same MSA.
Yes.
Then you're generating sequences, but then for some reason your model is adding some additional information that will be predicted.
Yeah.
Right.
Yes. Yeah. The POTS model actually, like, I think does quite well for this task. It seems that the EvoDiff model has like learned something about passing. Either my thought is like, because we're subsampling MSAs, we're only giving it 64 at a time. Over multiple epochs, it can like resample an MSA. So it's seen many examples of aligned sequences over smaller chunks of that sequence. Whereas a POTS model only gets it kind of that one time that you're fitting it to an MSA.
Maybe I'm just not correctly interpreting the plot.
Yeah.
So is upper left better or bottom right is better?
Bottom right would indicate more diverse sequences and that the average TM score is like about 0.5, whereas the POTS model is like on average just like regenerating the sequence, the query sequence. And so then it's giving a really high similar TM score. Does that make sense? Okay.
Yeah, I don't know if I'm being correct, but
Okay.
I'll ask you.
Okay. That's
Thanks so
fine.
much.
Yeah, of course. Okay. Okay. And so next we're going to move on to this tab that we've coined sequence and painting, which is effectively taking out a chunk of a protein sequence and trying to regenerate both with sequence alone and with MSAs. And to do this, we use this example of generating on intrinsically disordered proteins. These are proteins that exist in collections of conformations and lack a distinct 3D structure. But we know that these proteins are, overlooked, but facilitate a large amount of these biological processes that are totally distinct in mechanism from their structured counterparts. We know that they play a central role in molecular recognition, as well as cellular regulation. Oh, sorry. And so, that's an example of something without structure. It just kind of wiggles around. It doesn't have a classical 3D shape. And just to remind you, when we generated some unconditional examples earlier, we saw that our EvoDiff-seq model was quite good at generating kind of this diversity of secondary structure, but methods like RF diffusion could not generate unstructured proteins. It's quite hard for these models to do that.
The variation in the, so in this model here on the left, how are you modeling the unstructured uses? And how is it that you know that this counterpart is structured?
Okay. So this is purely a schematic that I'm showing on the left of an example of something that's intrinsically disordered. But a good question here is like, what is the data set that we're using for this? And to do that, I'll just pull this up. But another one of our collaborators published a paper on combining sequences of various species according to their shared intrinsically disordered domains. And so they were able to create a data set that contained aligned sequences that were aligned by their shared disordered regions. And they could do this by aligning protein sequences via their shared structural regions. And so that's a way they were able to do this to curate a data set, if that helps. Okay. Very cool. And so, okay, for this example, we're doing this task both using our EvoDiff-seq model, so a model that's only seen sequences, and our EvoDiff MSA model. As described, we kind of do that same task as earlier, where we will mask out the query IDR region. And with the single sequence model, we just take that sequence and mask out the known disordered regions, and then task our model with generating new disordered regions. We can then pass these now generated sequences through a disorder predictor. These are not as well validated as something like AlphaFold, but work fairly well for our task here. And this gives us a readout of a predicted disorder score for every amino acid in the sequence, where high score is high disorder. And here just, this is one example from our EvoDiff-seq model of regenerating a disordered region that scores quite well. And similarly, we can do the same thing with EvoDiff MSA. Now, if we just look at the overall disorder scores, we wanted to do some benchmarking with true disordered regions, generated regions, as well as random regions. And then we see for both disordered and structured predictions, this is kind of a reliable predictor that we can use for this task. Now, interestingly, when we generate IDRs with our EvoDiff-seq model, we see that they're overall less similar to the original IDR, meaning we can generate things with more diversity. Whereas our EvoDiff MSA model, we generate things that are closer in similarity to the original IDR. And that makes sense because it's been past the context of this sequence alignment.
Last slide, what is random?
Random, we randomly sampled from known IDR distributions.
So.
Yeah.
The aim of this is to generate sequence that are predicted to be disordered.
Mhm.
And then the evaluation is like how disordered it is for the sequence you generated. And the higher it is, the better it is.
Correct. A higher score indicates more disorder.
Would it be just like run scramble analysis?
Ah, yes.
Yeah, because I would imagine it could be quite disordered.
Yes, we did that analysis. I think we have a plot in the SI. It was different enough.
Yeah.
But yes, you're right. Yes, you're right on the scrambling and IDR makes sense for evaluating this task.
Questions like if your last slide was about the disorder regions play important roles in biological functions.
Mhm.
I wonder if you consider like generate IDRs that actually like actually evaluate this generate IDR based on their likelihood to preserve biological functions.
Yeah, that would be so cool. The question comment is like, if we can evaluate these IDRs for preserved functions, specifically the ones we're generating, and yeah, that would be quite cool. It requires access to like experiments, and we're like very much just a dry lab, so.
Well, like for example, some disorders, short disorder segment bind to structural protein, and you can evaluate how well your generated short segment bind to the science protein.
Oh, like a peptide binder.
Yeah.
Yeah. Yes, yeah. We could totally, yes, we should chat more. I have a separate project that may be of interest to that, where we've been designing short peptide binders that are for all, thoughts of ours that are disordered, so. But yes, yeah, that would totally be a valid use of that. We should do that with this, yeah.
I confirm something
Yes.
trivial, which is, when you say disordered, you don't mean simply not strand, not helix, you mean disorder.
In this case, I mean simply not stranded, not helix. I'm not, just like, saying it's like a coil or a loop or,
You don't know if it's.
yes, exactly. Yeah.
Separate thing. Okay.
Yes, correct.
I guess to clarify, the disorder score here is active in the secondary structure. Correct, look at not.
So the question is like, basically, what is this disorder predictor? We pass it through a model called DISOPRED, which is particularly aimed at predicting disordered regions. Yeah, if someone who better understands the definitions wants to speak to that, welcome you to speak now. But my understanding is like a disordered protein has a very particular combination that is unique from just a coil in a protein that might wiggle around. So it might be like a particular part of the sequence that enables a new conformation. So versus just like a coil present in a structured protein that might just like fluctuate. Does that make sense? Are there any strong disagreements against this definition? Some people who work on disordered proteins feel very strongly about these definitions. Okay. Yeah, and if you do have thoughts, I would love to hear them, and please find me. Okay, and so for our last task, we call this functional scaffolding, which we steal from the protein structure world, which is based on this idea that many biological functions are mediated by these motifs, which are then stabilized by scaffolds. We see this in designing various epitope binders, these viral receptor traps, active sites, or just protein-protein binders. And now we really kind of wanted to ask this like bold question: can we scaffold structural motifs just using sequence information alone? And it turns out that we kind of can. So what we do is we take a protein sequence. These are taken from RF Diffusion's benchmark that they published a little while ago. We pull out that functional motif that we're interested in scaffolding. we randomly place it in a protein sequence of kind of a range of sizes, depending on the task that is benchmarked. And then we iteratively decode the amino acids that scaffold this motif. Like so. And at the end, we can now get a folded prediction of what this resulting sequence is. In this case, we got something that matched the original sequence quite well. And we're able to do this without any structure, which was like quite mind-blowing to us. And we can do this for a number of various functional motifs. So here's an example that I just showed where we scaffold one PRW. Here's another example that we scaffolded. This is an active site of the carbonic anhydrase metalloenzyme. And overall, we see that we can solve on par the number of problems as RF diffusion using our EvoDiff MSA model. The EvoDiff model does like quite a bit worse. It only solves 8 of the 17 problems. Overall, we have like fewer numbers of successful scaffolds. These are not experimentally validated. Go for it.
Do you need to keep constant the number of positions between the constant regions?
Yeah, this is a great question. This is something we're playing with now. For these examples, we kept it fixed because there was no like reason in our heads to kind of complicate this as a benchmark for sequence, but it would be fun to like see if it can like retain these, especially these really small motifs if we place them much further apart. Yeah, oh.
Yeah, like you took 3 CDRs and asked it for a scaffold, does it give you an antibody?
I have not done this. There is a paper on bioRxiv somewhere. I believe the author's name is Colby, and I think he tried something similar to this. I don't think he tested them, but I think the designs are out there, so you could go find them. Colby, I'll remember his name. I will find the paper. Cool, okay, and so now these points are representing those sets of kind of benchmarks that RF diffusion put out there. Each of these is a unique PDB, and on each axis, we're counting the number of successful generations from our seq model and our MSA model, and on average, we see that the MSA model does like quite a bit better than the sequence model. Similarly, we compare our generations against those from RF diffusion, the numbers that they report in their paper. And interestingly, we see that these sequence and structure-based models, going back to this question from earlier, have quite orthogonal strengths. So problems that we do good on can be quite difficult for RF, and some of the ones that they do well on are quite hard for us. There's a few cases where EvoDiff-seq does best. And in these, we found that these examples didn't have many homologs to construct an MSA, so it makes sense that that's kind of a harder task for the MSA model. But similarly, if we compare the generations from EvoDiff MSA to EvoDiff-seq, on average, we found that our seq model produced scaffolds that were typically more novel in structure. Cool, and so today we've showcased this model EvoDiff, which has attempted to controllably generate protein sequences via a discrete diffusion model. Shown you we can unconditionally generate new proteins. We've shown that we can conditionally generate a number of new examples, which we're actively testing, again, hoping to get results on these soon. But overall, we're really interested in this idea of kind of defining a function and generating new sequences and thinking of ways that we can progress towards that. To close it out, I just want to highlight the team that I work on. I'm with the biomedical machine learning group at Microsoft Research. We're just around the corner. And it's a team of really great people who are working across all types of problems in biology. I work on, molecules and just protein engineering, but there's people who work on cells, tissues, and patients as well. They're all great, and we always, if there are any students here, we take interns every year. Applications normally open up at the end of the year, like between October and December. So please apply if you're interested in any of these areas. And with that, I'll take, I guess, any more questions if there are any.
And thanks. Let's say that you were interested in designing new like active sites. I'm wondering if it would be an interesting exercise to do the inverse of the scaffold where you showed.
Yeah, I think if you're just substituting a few amino acids, there might be models that do better. I would guess like ESM is probably better at that task if you're, just replacing a few things, but it would be interesting if we're able to do that as well. Yeah. Thanks. Oops. I think a mic is being passed around, but I see you.
Can you explain why you chose to, or maybe have to use the order agnostic autoregressive model for conditional generation over the D3 model?
So there is no clear direct way to generate a conditioned protein sequence with D3PM. You may need to like additionally train that model for that task. There's no reason we couldn't do it, though, and we probably should. That would be like very interesting. The model just didn't do as well as the order agnostic diffusion. I know there's some works out there that have used there's a D3PM absorbing model, which masks a set number of residues at each time step that is not one step equals one masking of one amino acid. I don't know if they've probably done some like, training of the model to do conditioning, I would imagine.
Thank you.
Yeah. I think I'll get your question.
I wonder how sensitive is your model to the length of the motif? Like, can you go with very short motif, just a few amino acid, or you have to with, like 10 amino acid each and several pieces to form a functional motif?
Oh, in the scaffolding task? It's very interesting. It seems not super related. So like, here's an example where we're giving it a very few things
That's pretty impressive.
spaced apart. There are other examples like this where we don't do a good job on. So it seems more related to like the type of sequence and like potentially it's like presence in the data set would be my guess, and the type of the like frequency of seeing this motif in the data set. I need to think about this more though. Oh, yeah, sorry.
Do you think your method is applicable to generate like new antibody mutants that like take some therapeutic antibodies and generate a bunch of variants and see how they will?
Yeah, I'm sure there's people out there using it. I know of like one paper, one offhanded example that I mentioned earlier where they're trying to do that, but Microsoft is like not super interested in making antibodies, so it's like not something that we've explored, but I welcome other people to do it. Yeah. Okay.
Yeah, so I'm sort of following up on a question that I asked earlier. And I'm just trying to understand how the model actually works. So
Yes.
So you mentioned that during training, you mask out a bunch of positions.
Mm-hmm.
But then one training step is you have the model predict all the masked positions.
Mm-hmm.
So then how do you use it in inference? Like, what does a decoding order mean?
Yeah, I
You
did.
have to add the ways back or remask things, you unmask and remask.
Yeah, we could do that. The simplest decoding order, because it's like learning from this. Okay, how do I? This maybe isn't the best graphic, but effectively we're learning a time step from 1 to length of sequence that is many decoding masks. So at time step 1, only one thing is masked. At time step 500, everything is masked. And so then to decode, we iteratively unmask one at a time. Like it's still ordered in the masking operation, even though we're doing it in an order agnostic way.
I guess I'm just wondering how it's different than, say, what protein amino, which is just an autoregressive sequence decoder. which essentially you start with no, in an inference, you start with no
Yes.
sequence. You choose a decoding order, you decode residue, whatever, and then you condition the next random sequence in the decoding order on that residue. You keep
Do you-
going until everything is.
Do you have to condition at every step of decoding in that way? Okay, so we wouldn't have to do that. No, because we've learned every step.
It just seems like the exact same output.
Okay, it possibly is.
It might be.
It might be.
Which is, I just don't know why it's diffusion as opposed to progressive.
Yeah, this is, yeah, it's the mathy way that they describe this in the original paper, which I can send you, but it's probably a version of the same thing. It's a Hooch Boom is the author.
But you're not, just to be clear, so you're not predicting all the mass tokens and then just taking one of them and then fixing that and then predicting all the mass tokens in the next step. Or I guess
I'm
I'm just
not.
not clear what the actual.
Okay,
We can talk about it.
yeah, let's chat. I think I can clarify this hopefully.
You mentioned that there were different, it was kind of mutually exclusive which proteins RF diffusion and EvoDiff MSA were successful on. Do you have any ideas about the different characteristics of one versus the other being successful?
Yeah, no, I was trying, I spent a very small amount of time investigating this. I have not found a satisfying answer, I don't think. There are some that we both do well on for some reason, like 1-BCF, it's a super helical protein. But there are other helical proteins that we do bad on, so it's not clear to me why this is orthogonal.
Yeah, great talk. I'm curious about the practical implementation, like how long does it take and how does it scale with sequence length?
Yeah, inference is one amino acid at a time, so it's quite slow. In the OADM paper, they propose that you can like generate many at a time to speed it up. I did not when I implemented it, so it was like quite slow, but there's certainly ways to make it faster.
Slower than like our diffusion.
I have not done this of like a time to, yeah, I have not done a one-to-one. That's a good, like a compute time. Yeah, that would be a good comparison. Wait, I saw that hand first, I think, all the way in the back.
It's like, it's the training set of sequences look like in terms of the protein family representation and how you choose the proteins to validate or, I don't know, these various results on like for the functional motif, for example, I would have just seen.
Yeah.
Or like something out of the sky.
Yeah. No, this is a good point. So we have a few different test splits. One is just a random test split that's done evenly. We have another harder split that Kevin generated that is aimed to be towards functional families, and we do a little bit harder. We're a little bit worse on that one, but it would be, yeah, I think the thing to investigate here is definitely on this maybe the orthogonality here is due to that presence of similar families in training. That would make perfect sense, yeah. Yeah, oh, sorry.
That was my question.
Oh, cool. Glad we're all on the same page.
Thank you so much. So you showed us a lot of different ways that you evaluated your model, such as the self-consistency complexity and some of the other graphs that you showed us. But at the end of the day, I haven't tried using the model, but when it spits out a list of sequences, is there a ranking in terms of how successful? Does it give you a sort of prediction on which ones will be more successful than others, or is it up to us to test them?
Yeah, no, we haven't implemented that. I think that's a really cool, good idea to have some sort of metric. We've been using these external codes that are available. Yeah, it would be fun to think of some like way of ranking.
Some kind of scoring metric that could take into account everything.
Yeah, that would be quite cool. Yeah, thank you. That's a good idea.
Are there
high throughput physical validation methods available to you?
Physical and,
Meaning the, you know.
oh, like experimentally.
Protein, you see if it,
Yes.
it falls.
This is what we're in the process of doing this with like a really small subset. Yeah, hoping to have those out when the final paper is out, hopefully. Yeah. But yes, this is something we're testing, but we're very limited in what we can test, which is frustrating.
Yeah, I was wondering if you could apply more high throughput methods that would at least monitor the amount of alpha helical.
Yeah, that would be like quite cool.
And that was consistent.
Yeah,
And
this is
that
true.
might get back into your rank.
Mhm. Oh, in the back.
Yeah, so in the first task that you described, you're essentially providing the model a bunch of list of functionalities that you want the molecule to do, and then it extends that you want the molecule to do, right? Like the protein.
In the generation task?
Yeah.
Yes, we provide a pixeling. Yeah.
So, it's a randomizer, but have you played around with having kind of something equivalent to a US token there that sometimes you may provide it with and you want and the model is that the story is disordered.
Oh, like if it could learn length. Yeah, if we probably add it, yeah, we did not use like a start stop token. So like we could probably implement something like that and have it learn, but we did not do that.
Okay.
Oh, whoops. Sorry. Yeah. I see, yeah.
Yeah, thank you for the nice talk. So I have a very simple question regarding to the sequence length. So if the position, like the position of amino acid is associated with time step, does it mean like during the training step, you have to group out different lengths of sequence, or do you have to specify length of sequence before you want to generate something?
You have to specify them. So during training, the time step is via the protein length. The lengths of proteins within a batch do not have to be fixed because we can sample that per protein. And at inference time, we do have to decide how long we want the protein. We'll have it like fill up to some amount of
Okay, thank you.
positions.
Hi, I have a question related to the scaffolding capabilities of this model. Can this model scaffold, for instance, active sites that have like a cofactor coenzymes?
It has not been trained on anything that's not a protein sequence. You could certainly try it on a cofactor enzyme, like pick out some amino acids and try to scaffold it. I don't know how successful it would be. The benchmark that we use is like quite small, so it's certainly like not sufficient to claim robustness, I don't think, but.
Yeah, I was wondering just because I use that providing pathways.
Yes. Yeah, we don't include anything about like an ion or anything when we generate it. It's just the protein sequence. Yeah.
I have another question about functional scaffolding. So you mentioned that there are a lot of examples that's failing to generate. I was thinking that one of the reasons might be that the sequences of those like functional scaffolds will fall depending on the context. So maybe the like context that's being put in that causes the sequence to fall in different fashion. So I was thinking that maybe one method to kind of circumvent this is to try maybe different like different sequences that falls into the same motif, maybe like homologous sequences from different proteins, which may be able to like sample different like sequences so that we can maybe some of them will be successful. I don't know if that's
that makes sense, like using a homologous protein as the scaffold versus the protein itself. Yeah, that could potentially work. Yeah.
Well, let's thank Sarah again, and more questions.